continue our discussion on gene expression. So now we're going to go back and look at the other end of the central dogma. So by now you should know, and you should be able to give me a definition of what the central dogma is in your own words. And we're going to look at what is the relationship between DNA and proteins. And this is something else that by now you should be able to address efficiently. So translation is the second part. So we have created our mRNA molecule during transcription, and now we're going to create a polypeptide during translation. So with a little bit of history is Archibald Garrett in the 1900s, he was an English physician, and he proposed the idea that genes encode for proteins. He didn't necessarily do an experiment, but he was studying a disease at the time, alkaptonuria. And he was saying, I think that the genes encode proteins and there's one missing, and so therefore these people have this disease. And so this is the metabolic pathway of interest and related to his disease, and he described the disease as an inborn error of metabolism. He knew it was autosomal recessive, but he didn't necessarily understand exactly what was going on at the molecular level. So then Beadle and Tatum came along, and they said they had a question to answer. Does one gene control the production of one enzyme, or does one gene control many enzymes in the same pathway? And so to figure this out, they did an experiment with neurospore, and they had different mutant strands of it. And what they wanted to do was have a mutant strand and then supplement it to see if they would get growth or not. So if strand 1 here was missing enzyme 1 needed for the conversion, then it can only grow if that was supplemented. Okay? So they would knock out that strand, all of them here. So knock it out and then supplement it, it grows. Knock it out and supplement it and see what happens. And so from this, they said that there is a one gene, one enzyme hypothesis. So they're saying that one gene codes for one enzyme. Is that correct? Is it accurate to say that one gene codes for one enzyme? So take a second and ponder on this idea. Does one gene code for one enzyme? Well, at the time, this was considered correct. But now that we have more data, more experiments, we know that that is not quite the truth. So all proteins are encoded by genes. So it's not one gene, one enzyme. So how about one gene, one protein? Would that be accurate? No, because some proteins are more than one polypeptide. So to this end, what is the difference between a protein and a polypeptide? Well, a polypeptide is going to be that amino acid sequence. It's just sequential. A protein is the functional unit. So usually a polypeptide has to be altered or modified before it's a functional protein. Then we know one gene can encode more than one polypeptide. So if you think back to the lesson we just had, you should be able to name 3 ways that this is possible. And then genes don't always encode polypeptides. They could encode functional RNA molecules. So one gene, one enzyme is not accurate. What about one gene, one polypeptide? Would that be an accurate statement? So now let's go through some things that you need to know to be able to understand translation. One is the genetic code. So when we talk about our genetic code, we're talking about we have our DNA, and from that we make our RNA. This RNA is broken down into 3. So our genetic code is a triplet code. 3 codons on an mRNA molecule code for 1 amino acid. Okay? And this is our genetic code. This is the language of proteins. So here you see it again. These are our codons. There are 64 codons in our code, each in triplets. There are 20 amino acids. So that tells you there that we have a degenerate code, and we'll look at that in detail. To read this table, you take the first letter, the second letter, the third letter. So if I present you with a sequence and I say, tell me what does AAU code for, you're going to go AAU, ASN, okay? That's how you read the code. So it's degenerate because we have 64 codons and only 20 amino acids. So that means that some codons code for the same amino acid. That's the degeneracy in our code. Those amino acids that code for those codons that code for the same amino acid are considered synonymous. So synonymous codons code for the same amino acid. UUU, UUC. Those are synonymous codons. They both code for phenylalanine. Because this is in our code, it's considered degenerate. Here's our DNA, the mRNA strand, the polypeptide sequence. This is codons are sets of 3. We have a start codon, which is AUG. It codes for methionine. Then in between the start and the stop, you just have codons. Codons exist on the mRNA sequence. You have a stop codon. There are 3 of those in our code: UAA, UAG, and UGA. Anticodons exist on the tRNA, and they are complementary to the codons on the mRNA. We have a 5' untranslated region on the mRNA. What does that mean? There is an area at the front or in the 5' that doesn't translate into a protein. There's also an area at the end. And this should be almost a no-brainer because remember, we capped this and we tailed it, and those areas are not going to be translated as well. And so then from start to stop is where we have our codons. Start codes for an amino acid, stop usually does not. We have something called wobble base pairing. So remember that there's 3 codons, and you want this to be as exact as possible, but it's possible in this 3rd one that it can wobble or allow an incorrect base pairing. So why is that? We'll talk about it in another slide where I have a picture so you can understand wobble. Now, the reading frame is sets of 3, but how do you know which set of 3 to start with? Okay, because if you look at the same sequence, if you start at the first letter, you get valine, the second, serine, the third, histidine. How do you know where to start? The answer is usually, where's the start codon? So here's our start. So we're going to start. We're going to allow the start codon to establish the correct reading frame. Our genetic code is almost universal. Almost universal. Okay? So there are some exceptions here, but usually most of them, all living things, use this code. The exceptions being what some things code for. So AUA is usually isoleucine, but it can be methionine in some organisms. These are our stops under normal circumstances, but in rare circumstances, UGA can be selenocytosine and UAC can be pyrrolysine. These have been called the 21st and 22nd amino acid by some people. We have our peptide bonds. So the carboxyl end of one amino acid bonds to the amine end of another. We lose water, we get this peptide bond. Peptide bond joins amino acids together. This is a polypeptide chain, many bonds. Okay, so we see here is our mRNA sequence. Here is the amino acids we would get from it. Our amino end is at the 5' here. Our carboxyl is at the 3'. Every 3, we get an amino acid, peptide bonds in between them. These R groups, so you have chemistry, you should be familiar with R groups by now, determine the nature of our protein. So here's our 20 amino acids, and we know that they have different characteristics. So we have some that are charged, positive and negative, some that are polar, some that are not polar, some are acidic, some are not acidic, and depending on this, it's going to help it to fold later on. So that brings us to the levels of protein structure. The primary is going to be the sequence of amino acids connected by these peptide bonds. Secondary can be alpha helix or beta sheets. Who discovered the alpha helix? You should be able to answer that. This is largely held together by hydrogen bonds. Then you form this into a 3D shape. This is the tertiary. This has different interactions that hold it together. A lot of protein stop here at the tertiary structure. However, if a protein is made up of more than one polypeptide, then it's a quaternary structure. Okay. And we will stop here and pick up on tRNA and rRNA in the next